Media presents Introduction to Biotechnology, a videoactive presentation. The goal is to offer an overview of some of the capabilities and potential of biotechnology. The objectives are to understand basic principles of molecular biology, to understand some of the applications of biotechnology in plants, to understand some of the applications of biotechnology in animals and medicine. Biotechnology is a rapidly expanding field. Scientists are continually learning.
To manipulate genes and gene products in medicine and also in the field of agriculture.
Depending upon the imagination of the people that are involved, there are so many things that could theoretically be done.
New technologies are advancing so quickly that in a very short period of time we should be able to transfer a gene from one animal into another with relative ease to affect things like human health, animal performance, survival in hostile environments, etc.
The smallest unit of life capable of independent existence is called a cell. The genetic instructions needed to make an entire organism are located in chromosomes within the nucleus of a cell. Chromosomes are made of a molecule called DNA. Each specific building instruction of an organism is recorded as a gene within the DNA. Genes control traits within the organism, everything from eye, hair, and skin color to inherited disabilities and diseases such as hemophilia, cystic fibrosis, or cancer.
So genes are responsible for the differences between individuals. The corn on the left is genetically different from the corn on the right. They're the same species. They're the same corn. They just differ in one or two genes, much as people with different eye color or skin color can differ in just one or two genes. And this diversity is critical for plant breeding because it's what provides the genetic basis for selecting plants with new traits.
In nature, there are two ways life reproduces and passes on genes: meiosis and mitosis. Fertilized eggs undergo mitosis to produce a completed organism. In mitosis, body cells duplicate. This is how virtually everything in the human body develops and is maintained. In plant tissue culture, mitosis can be used to produce cells with the capability to generate identical clones from the cutting of a parent plant. Meiosis results in cells having only half of the required set of genes. During fertilization, a male haploid cell, or sperm, fuses with a female haploid cell, or egg, to produce a cell that has a complete set of genes. This is called sexual reproduction, and it occurs in nearly all multicellular organisms. If a breeder wants two particular traits to appear in the same organism, the breeder would breed two organisms together, each parent having one of the desired traits. For example,
if a breeder wanted to transfer some of the stress tolerance from this line of corn to this, then pollen from the tassels could be transferred to the silks on the ears of this corn, and then the offspring could be selected for increased stress tolerance.
Distantly related organisms cannot breed. Under natural conditions, crossing genes of an animal and a plant is impossible. But biotechnology and transgenics are offering new tools to scientists.
What they would be very interested in and inserting into grapes would be genes for cold hardness, if those could be found. There are fish that survive under extremely low cold temperatures, and those genes have been taken out of fish, and there's interest in putting those into various crops to see if they also can confer cold hardiness in those crops.
When an organism receives a new gene, such as a great plant receiving the fish gene, that organism is said to be transformed or transgenic.
Transgenics can be simply explained as the transfer of a gene into another organism, whether that be taking a gene from one bacteria and putting it into another bacteria, or a gene from one mammal, such as a cow, into a different mammal, like, let's say, a pig. When we add transgenic genes to bacteria or single cells, such as a mussel, we have no way of telling if that mussel cell or that bacteria is different from a normal bacteria, because a transgenic cell will look just like a normal cell. So we incorporate what are called marker genes. Marker genes are genes that are incorporated into the animal to visually identify a transgenic animal. They can be everything from coat color, fluorescence, and antibiotic resistance. And we talk about single cells or bacteria, that's usually a fluorescent marker. When the gene of interest and our marker gene are both incorporated into that cell, it'll show a fluorescent color, be it green or red. That is a protein that's been isolated from a jellyfish. So we've taken jellyfish gene and put it into a pig cell or bacteria cell and had that jellyfish green fluorescent expressed in a non
ways life reproduces and passes on genes. A. Replication and duplication. B. Mitosis and meiosis. C. Plant culture and animal culture. D. DNA and genetics. For natural sexual reproduction to take place, parents have to be A. The same size. B. The same color. C. Of similar species. D. The same age. Mitosis is used A. In plant tissue culturing. B. To grow and develop tissues. C. All of the above. D. None of the above.
Genes for specific beneficial traits can be isolated and inserted into a plant species, creating a transgenic organism by adding to its genetic code.
The most commonly used method is agrobacterium.
Ordinarily, this bacterium infects plant cells by injecting some of its genetic material into a cell and using the cell to create its food.
We've replaced the bacterial genes with the genes we want to put in the cells.
When these bacteria infect cells, they transplant the gene of interest into the cell's chromosomes among the cell's original genes.
There are mechanisms which aren't understood that get the DNA from the bacterial cell into the plant cell, and once into the nucleus, into the chromosomes. I'd say the overall goal of plant biotechnology is to improve agriculture. Whether that means through reducing the costs of growing, allowing crops to grow under more marginal areas, increasing the quality of the crops.
Some agricultural qualities scientists would like to enhance in plants include resilience, herbicide resistance, harvestable quantity, flavor, nourishing quality, and spoilage resistance. There are many benefits to growing crops in places where it was previously not possible.
We're actually doing grape varieties that are tolerant to salt, which is a big issue for areas in which they grow a lot of grapes that have limited water. The water that you do have may be very salty. It may be very low quality water, yet you could still be able to grow the grapes that are very good and of high quality.
As agriculture gets pushed further and further, we're going to have to use more marginal areas, areas that maybe are traditionally too dry.
If genes could be found that defend a plant from insects, fungi, viruses, and bacteria, then losses could be greatly reduced.
Already existing in nature are thousands and thousands of compounds which for as long as we have been growing plants have conferred to those plants some selective advantage. What we would like to do is to design plants with those compounds to meet our needs. For instance, cotton plants, grapes, most vegetables, tomatoes, carrots, in this case onions, all of those are susceptible to soil-borne pests, which when they attack them, they can either kill the plant or they can render it so that it's useless horticulturally, and then we can't eat that. There's a corn called BT corn. There's also BT cotton, and BT stands for Bacillus thuringiensis, a soil-borne bacterium. It was found when insects fed on this, they died. Well, if you can incorporate into the leaves themselves this BT gene, then what happens is the insect ingests protoxin and the larva dies. This means the growers don't have to spray their fields.
I read an article this morning about pesticide use in China on BT cotton, and they've been able to go from 15 pesticide applications to 1 pesticide application a season, which is incredible.
So it's cheaper in the long run, it's much better for the environment in the long run, and it is good for the consumer.
If we don't use BT, then the farmers are going to have to spray with conventional pesticides. In a few weeks, there'll be a bloom, an increase in aphids. Those conventional pesticide applications will kill the beneficial insects, the insects that eat the aphids, things like ladybugs.
Some plants are engineered to be immune to herbicides. This enables the farmer to apply herbicides at more opportune times, increasing production.
Farmers using herbicide-resistant crops can do much less cultivation, and instead of controlling the weeds with mechanical cultivation, they control them with extremely safe herbicides. Where before farmers would plow a field, plant the crops, and then have to cultivate that field to get rid of the weeds. And maybe 4 or 5 times they'd have to drive a tractor burning diesel fuel through the field, compacting the soil, leading to more soil erosion.
Reduced mechanical cultivation means reduced fuel usage, reduced soil erosion, and increased water efficiency. A key concern is meeting the agricultural needs of an increasing world population. If more can be harvested from the same amount of land and water, humanity will be better equipped to address this concern. Cotton is undergoing research in an effort to increase the amount of cotton fiber produced from each plant.
If you can engineer or you can somehow select plants that have higher rates of photosynthesis, again, you can increase productivity on a given amount of land for a given amount of input, of water and fertilizer input and pesticide input. And some of these lines you can see have a large number of flowers. Here's a large number of flowers and a large number of buds, which is good. Each flower has the potential to result in a boll, and the boll is where the cotton is formed. So more flowers can mean higher yield, more productivity.
Some plants can be genetically engineered to produce fruits and vegetables with more flavor and better color, increasing the enjoyment of a food.
The reason that when you go to a grocery store and buy a tomato and cut it open and take a bite, it tastes like sawdust is because to be harvested by machine, they need to be hard and firm because the machines can't be gentle with the tomatoes. They have to be harvested green so they don't over-ripen by the time they get on the grocery store shelf. By introducing these genes that slow down the softening, you can harvest the tomato later after all the flavors have developed, but they'll remain firm and resistant to spoiling longer on the shelf.
This enables crops to be shipped further distances, stored for longer periods of time, and picked closer to ripeness, decreasing spoilage. Increased flavor is accompanied by increased nourishment.
Now, grapes are an example of a crop that is just packed with the antioxidants that we have heard a lot about lately.
In underdeveloped parts of the world where nutrition is lacking, a staple food can be engineered to supply missing proteins, vitamins, minerals, and other compounds, improving human health.
Rice traditionally is very, very low in vitamin A, and in fact it's responsible for malnutrition leading to blindness and death in many poor countries where rice is the staple food. And golden rice is rice into which genes for the production of vitamin A have been introduced.
There is even work to make vaccines in plants, making immunization against diseases in underdeveloped countries more efficient and less expensive.
In the third world countries, they don't have access to many of the medicines that we have here. And there's a real need to provide for them vaccines.
If, on the other hand, you could ship a batch of bananas that carry the vaccine, that express the vaccine in the fruit, you can feed the kids the bananas and they become vaccinated. This kind of technology certainly could save millions of lives in the third world where childhood diseases, diarrhea, bacterial diarrhea and things like that kill literally millions of kids every year. If you could vaccinate them by just eating a piece of potato or a piece of carrot or a piece of banana, it would be incredible.
Advances in agriculture offer more solutions to commercial farming businesses. By enabling more possibilities, humanity is better equipped to address concerns of nutrition, hunger, health, and well-being for an increasing population.
Quiz 2. Goals of biotechnology in plants include making plants A. Stronger, B. More productive, C. More nourishing, D. All of the above. Rice is usually very low in A. Vitamin A, B. Carbohydrates, C. Starch, D. All of the above. Biotechnology may help some people in developing countries who need A. Vaccinations and disease prevention.
Though it is very difficult and costly, an entire organism can be generated from a single cell taken from another animal.
Cloning of sheep is a very important issue in animal agriculture for one primary reason. If we can get an animal that makes a large amount of wool, is a very high quality muscled animal that would help solve our needs of feeding the hungry people in the world, one of the best ways would be to copy that same animal over and over and over again. Pig cloning was originally started as a way to make animals that had very similar meat quality traits and performance traits so that the expense of managing the pig could be minimized. However, scientists quickly realized, because of the similarities of the pig to the human physiology, that there was a possibility to clone pigs for human organ transplant. Pig tissues and organs are currently being used in human medicine. For example, they currently take pig skin tissue and actually place it onto severe burns in a human and repairs that skin. They actually currently transplant pig heart valves into humans. There are some people that have pig arteries and blood veins in their legs and arms that have repaired damaged arteries and veins. Current research is looking at using pig livers and kidneys to repair liver damage and kidney damage in humans. Cattle, sheep, and goats don't make good candidates for human transplant, organ and tissue transplant for one primary reason. They're entirely different than us. Their organs are of a different size, located in a different place, and actually perform slightly different functions. The cow has been heavily studied both in normal agricultural production and transgenics and cloning. We have a significant amount of data and can do cloning relatively easily in cattle compared to the other three species that we've talked about. I think in 10 to 15 years, cloning sheep, cattle, or pigs will be extremely affordable, and everybody will have the ability to do it, which will decrease the cost of producing animals, producing organs for transplant, producing transgenic compounds for administration to humans for disease. So I think cloning has a huge potential to improve the overall quality of life of many people around the world.
By inserting genes, animals can be transformed into bioreactors, living factories of important chemicals.
Sheep and goats make an excellent bioreactor system because they have a very large and productive mammary system. It allows for us to add a human gene to their genome, allowing them to produce the protein from that human gene into their milk. Cattle are also used as bioreactors. Again, the primary advantage to cattle is they have a very large mammary system and can produce thousands and thousands of gallons of milk per year that you can isolate a humanized protein out of. But a problem with cattle, as with the goats, is that it's hard to get human genes into these animals to produce human proteins. The reason pigs have become a candidate for bioreactors for human compounds is because of how closely and similar their organ, tissue, and physiological systems are to those of humans. Pig proteins look a lot or are very similar to human proteins. And so while researchers have used larger milking machines like cattle and goats, they've come back to the pig because the proteins that they produce very closely resemble the proteins that a human produces. So when we isolate a protein from their milk and inject it into a human, there's a far less chance of an immune reaction to that than there would be from cattle or goats or sheep, because the pig has the similar reproductive, digestive, and overall physiological systems that humans do.
Perhaps a gene transferred into cattle will enable them to produce enough human blood clotting factor to treat patients with hemophilia. Or perhaps transplanted organs will be able to produce insulin and cure diabetics. But what about small animals, such as pets?
Every year, the United States spends something like $5 or $6 billion treating diseases in pets, in dogs and cats mainly. If we had accurate information on all the genes in cats and dogs, it would be possible to breed out these genetic defects that cost so much money and cause pet owners so much grief. There's a large number of people in the United States who are deathly allergic to cats. And this is mainly because of a particular gene in the cat that makes a protein present in the saliva of the cats when they groom themselves. And this is mainly responsible for the very violent reaction that a lot of people have against cats. Now, it's perfectly reasonable that we could find this gene and remove it from the cat genome. We could breed strains of cats that were not allergenic. And once again, you could look at a tremendous saving of money and grief. Lots of people want to have cats, but they can't have cats because they're so allergic to them.
Colonies of specific types of cells can be produced that ordinarily couldn't be produced so quickly. In the human body, lymphocytes produce antibodies which neutralize bacteria, viruses, and even cancers. These antibodies are key in the body's defense against disease. Cell cultures can produce large amounts of antibodies which can be tested, studied, and manipulated to treat diseases such as cancers or infections.
There's a lot of effort going on in terms of trying to understand disease processes on the molecular level. Cancer, heart disease, genetic inherited disorders. The DNA of the human genetic constitution has been read. That is, all the letters in the genetic alphabet are being translated. And it should help us in the long run to understand a lot of genetic diseases. If you understood them, you could probably design drugs and therapies that would act against a debilitated genetic constitution and would predict people who were at risk for heart disease, for strokes, heart attacks, and so forth. And autoimmune diseases are a huge problem in the United States. Rheumatoid arthritis, of course, is a big blockbuster disease. Diabetes, lupus erythematosus, Crohn's disease, Hashimoto's thyroiditis. So there's many, many different diseases. Autoimmune diseases are going to be one of the big frontiers of research in the coming years. They affect many, many people, and they're not well understood at all at present.
Quiz 3. Examples of biotechnology in animals include A. Animal cloning, B. Cancer research, C. Human tissue replacement, D. All of the above. A transgenic organism is one that A. has genes inserted into it from another organism, B. has genes created through mitosis, C.
Is not without its risks. As with all new technologies, there are controversies. Since biotechnology has such a broad scope, it has an equally broad range of concerns. we may be able to find the answers.
Final quiz. In humans, genes control all of the following except A. Nutrition, B. Eye color, C. Skin color, D. Inherited diseases. Simple cell division is called A. Mitosis, B. Meiosis, C. Sexual reproduction, D. None of the above. Biotechnology can be used in all of the following except A. Plants, B. Animals, C. Human medicine, D. None of the above. The most commonly used bacterium for gene transfer in plants is A. Bacillus thuringiensis, B. Escherichia coli, C. Agrobacterium, D. None of the above. When a gene is transferred into an organism, the organism is called A. Transgenic, B. Transformed, C. All of the above, D. None of the above. This program discussed vaccinating people through A. Fish, B. Bananas, C. Rice, D. None of the above. Of the following, which is most likely to increase a plant's productivity? A. Increasing its rate of photosynthesis, B. Decreasing its rate of photosynthesis, C. Maintaining its rate of photosynthesis, D. Eliminating its ability to flower. The problem with many pesticides is that they may actually A. Cause an increase in some pests, B. Kill beneficial insects, C. Be harmful to the environment, D. All of the above. According to this program, cats produce an allergen in their A. Fur, B. Saliva, C. Claws, D. All of the above. What are inserted into a cell's DNA to visually identify transgenic character? A. Marker genes, B. Cultures, C. Clones, D. Bioreactors.
Imagine you were alive back in the 1980s and were told that computers would soon take over everything from shopping to dating and the stock market. That billions of people would be connected via a kind of web. That you would own a handheld device orders of magnitude more powerful than supercomputers. It would seem absurd, but then all of it happened. Science fiction became our reality and we don't even think about it. We're at a similar point today with genetic engineering. So let's talk about it. Where it came from, what we're doing right now, and about a recent breakthrough that will change how we live and what we perceive as normal forever. Humans have been engineering life for thousands of years. Through selective breeding, we strengthened useful traits in plants and animals. We became very good at this, but never truly understood how it works, until we discovered the code of life, deoxyribonucleic acid, DNA, a complex molecule that guides the growth, development, function, and reproduction of everything alive. Information is encoded in the structure of the molecule. 4 nucleotides are paired and make up a code that carries instructions. Change the instructions and you change the being carrying it. As soon as DNA was discovered, people tried to tinker with it. In the 1960s, scientists bombarded plants with radiation to cause random mutations in the genetic code. The idea was to get a useful plant variation by pure chance. Sometimes it actually worked too. In the 70s, scientists inserted DNA snippets into bacteria, plants and animals to study and modify them, for research, medicine, agriculture and for fun. The earliest genetically modified animal was born in 1974, making mice a standard tool for research, saving millions of lives. In the 80s, we got commercial. The first patent was given for a microbe engineered to absorb oil. Today, we produce many chemicals by means of engineered life, like life-saving clotting factors, growth hormones and insulin, all things we had to harvest from the organs of animals before that. The first food modified in the lab went on sale in 1994, the Flavor Saver tomato, a tomato given a much longer shelf life via an extra gene that suppressed the buildup of a rotting enzyme. But GM food and the controversy surrounding them deserve a video of their own. In the 1990s, there was also a brief foray into human engineering. To treat maternal infertility, babies were made that carried genetic information from 3 humans, making them the first humans ever to have 3 genetic parents. Today there are super-muscled pigs, fast-growing salmon, featherless chickens and see-through frogs. On the fun side, we made things glow in the dark. Fluorescent zebrafish are available for as little as $10. All of this is already very impressive, but until recently, gene editing was extremely expensive, complicated and took a long time to do. This has now changed with a revolutionary new technology now entering the stage: CRISPR. Overnight, the costs of engineering have shrunk by 99%. Instead of a year, it takes a few weeks to conduct experiments, and basically everybody with a lab can do it. It's hard to get across how big a technical revolution CRISPR is. It literally has the potential to change humanity forever. Why did this sudden revolution happen, and how does it work? Bacteria and viruses have been fighting since the dawn of life. So-called bacteriophages or phages hunt bacteria. In the ocean, phages kill 40% of them every single day. Phages do this by inserting their own genetic code into the bacteria and taking them over to use them as factories. The bacteria try to resist, but fail most of the time because their protection tools are too weak. But sometimes bacteria survive an attack. Only if they do so can they activate their most effective antivirus system. They save a part of the virus DNA in their own genetic code in a DNA archive called CRISPR. Here, it's stored safely until it's needed. When the virus attacks again, the bacterium quickly makes an RNA copy from the DNA archive and arms a secret weapon, a protein called Cas9. The protein now scans the bacterium's insides for signs of the virus invader by comparing every bit of DNA it finds to the sample from the archive. When it finds a 100% perfect match, it's activated and cuts out the virus DNA, making it useless, protecting the bacterium against the attack. What's special is that Cas9 is very precise, almost like a DNA surgeon. The revolution began when scientists figured out that the CRISPR system is programmable. You can just give it a copy of DNA you want to modify and put the system into a living cell. If the old techniques of genetic manipulation were like a map, CRISPR is like a GPS system. Aside from being precise, cheap and easy, CRISPR offers the ability to edit live cells, to switch genes on and off, and target and study particular DNA sequences. It also works for every type of cell: microorganisms, plants, animals or humans. But despite the revolution CRISPR is for science, it's still just a first-generation tool. More precise tools are already being created and used as we speak. In 2015, scientists used CRISPR to cut the HIV virus out of living cells from patients in the lab, proving that it was possible. Only about a year later, they carried out a larger-scale project with rats that had the HIV virus in basically all of their body cells. By simply injecting CRISPR into the rats' tails, they were able to remove more than 50% of the virus from cells all over the body. In a few decades, a CRISPR therapy might cure HIV. And other retroviruses, viruses that hide inside human DNA like herpes, could be eradicated this way. CRISPR could also defeat one of our worst enemies, cancer. Cancer occurs when cells refuse to die and keep multiplying while concealing themselves from the immune system. CRISPR gives us the means to edit your immune cells and make them better cancer hunters. Getting rid of cancer might eventually mean getting just a couple of injections of a few thousand of your own cells that have been engineered in the lab to heal you for good. The first clinical trial for a CRISPR cancer treatment on human patients was approved in early 2016 in the US. Not even a month later, Chinese scientists announced that they would treat lung cancer patients with immune cells modified with CRISPR in August 2016. Things are picking up pace quickly. And then there are genetic diseases. There are thousands of them, and they range from mildly annoying to deadly or entail decades of suffering. With a powerful tool like CRISPR, we may be able to end this. Over 3,000 genetic diseases are caused by a single incorrect letter in your DNA. We are already building a modified version of Cas9 that is made to change just a single letter, fixing the disease in the cell. In a decade or two, we could possibly cure thousands of diseases forever. But all of these medical applications have one thing in common. They are limited to the individual and die with them, except if you use them on reproductive cells or very early embryos. But CRISPR can and probably will be used for much more: the creation of modified humans, designer babies, and will mean gradual but irreversible changes to the human gene pool. The means to edit the genome of a human embryo already exist, though the technology is still in its early stages, but it has already been attempted twice. In 2015 and 2016, Chinese scientists experimented with human embryos and were partially successful on their second attempt. They showed the enormous challenges we still face in gene editing embryos, but also that scientists are working on solving them. This is like the computer in the 70s. There will be better computers. Regardless of your personal take on genetic engineering, it will affect you. Modified humans could alter the genome of our entire species because their engineered traits will be passed on to their children and could spread over generations, slowly modifying the whole gene pool of humanity. It will start slowly. The first designer babies will not be overly designed. It's most likely that they will be created to eliminate a deadly genetic disease running in a family. As the technology progresses and gets more refined, more and more people may argue that not using genetic modification is unethical because it condemns children to preventable suffering and death and denies them the cure. But as soon as the first engineered kid is born, a door is opened that can't be closed anymore. Early on, vanity traits will mostly be left alone. But as genetic modification becomes more accepted and our knowledge of our genetic code enhances, the temptation will grow. If you make your offspring immune to Alzheimer, why not also give them an enhanced metabolism? Why not throw in perfect eyesight? How about height or muscular structure? Full hair? How about giving your child the gift of extraordinary intelligence? Huge changes are made as a result of the personal decisions of millions of individuals that accumulate. This is a slippery slope. Modified humans could become the new standard. But as engineering becomes more normal and our knowledge improves, we could solve the single biggest mortality risk factor: aging. 2/3 of the 150,000 people who die today will die of age-related causes. Currently, we think aging is caused by the accumulation of damage to our cells, like DNA breaks and the systems responsible for fixing those wearing off over time. But there are also genes that directly affect aging. A combination of genetic engineering and other therapy could stop or slow down aging, maybe even reverse it. We know from nature that there are animals immune to aging. Maybe we could even borrow a few genes for ourselves. Some scientists even think biological aging could be something that eventually just stops being a thing. We would still die at some point, but instead of doing so in hospitals at age 90, we might be able to spend a few thousand years with our loved ones. Research into this is in its infancy, and many scientists are rightly skeptical about the end of aging. The challenges are enormous, and maybe it is unachievable. But it is conceivable that people alive today might be the first to profit from effective anti-aging therapy. All we might need is for someone to convince a smart billionaire to make it their next problem to solve. On a bigger scale, we certainly could solve many problems by having a modified population. Engineered humans might be better equipped to cope with high-energy food, eliminating many diseases of civilization like obesity. In possession of a modified immune system, with a library of potential threats, we might become immune to most diseases that haunt us today. Even further into the future, we could engineer humans to be equipped for extended space travel and to cope with different conditions on other planets, which would be extremely helpful in keeping us alive in our hostile universe. Still, a few major challenges await us. Some technological, some ethical. Many of you watching will feel uncomfortable and fear that we will create a world in which we will reject non-perfect humans and pre-select features and qualities based on our idea of what's healthy. The thing is, we are already living in this world. Tests for dozens of genetic diseases or complications have become standard for pregnant women in much of the world. Often the mere suspicion of a genetic defect can lead to the end of a pregnancy. Take Down syndrome, for example, one of the most common genetic defects. In Europe, about 92% of all pregnancies where it's detected are terminated. The decision to terminate a pregnancy is incredibly personal, but it's important to acknowledge the reality that we are pre-selecting humans based on medical conditions. There is also no use in pretending this will change, so we have to act carefully and respectfully as we advance the technology and can make more and more selections. But none of this will happen soon. As powerful as CRISPR is, and it is, it's not infallible yet. Wrong edits still happen, as well as unknown errors that can occur anywhere in the DNA and might go unnoticed. The gene edit might achieve the desired result, disabling a disease, but also might accidentally trigger unwanted changes. We just don't know enough yet about the complex interplay of our genes to avoid unpredictable consequences. Working on accuracy and monitoring methods is a major concern as the first human trials begin. And since we've discussed a possible positive future, there are darker visions too. Imagine what a state like North Korea could do if they embraced genetic engineering. Could a state cement its rule forever by forcing gene editing on their subjects? What would stop a totalitarian regime from engineering an army of modified super soldiers? It is doable in theory. Scenarios like this one are far, far off into the future, if they ever become possible at all. But the basic proof of concept for genetic engineering like this already exists today. The technology really is that powerful. While this might be a tempting reason to ban genetic editing and related research, that would certainly be a mistake. Banning human genetic engineering would only lead to the science wandering off to a place with jurisdiction and rules that we're uncomfortable with. Only by participating can we make sure that further research is guided by caution, reason, oversight, and transparency. Do you feel uncomfortable now? Most of us have something wrong with them. In the future that lies ahead of us, would we have been allowed to exist? The technology is certainly a bit scary, but we have a lot to gain, and genetic engineering might just be a step in the natural evolution of intelligent species in the universe. We might end disease. We could extend our life expectancy by centuries and travel to the stars. There's no need to think small when it comes to this topic. Whatever your opinion on genetic engineering, the future